Hello, everyone. Also from my side, a very warm welcome. Before we proceed into this webinar session, please acknowledge the following disclaimer about Miltenyi Biotec products and services. Thank you very much. Now, for this webinar session, we will go over different aspects about the manufacturing of gamma delta T cells. We will start discussing several key considerations when setting up manufacturing for gamma delta T cells. Next, we will have a closer look at the workflow we have developed to manufacture particularly one gamma delta T cell subset, as well as how we can implement efficiently genetic modifications into these cells. Finally, we will have a look at some flow cytometry tools for analytical testing and phenotyping of these cells. And of course, we will have some time for questions. Before we jump into the key considerations for gamma delta T cell manufacturing, I would like to take some minutes to go over some biological aspects of gamma delta T cells, as well as the clinical landscape for gamma delta T cell therapy. So what are gamma delta T cells? Gamma delta T cells are known as unconventional T cells because they can express both innate and adaptive properties. They are a very small population within the total T cell compartment in the body. And the major subsets that have been described so far are the B delta 2, as well as the B delta 1 and B delta 3 gamma delta T cells. And each subset has different tissue prevalence that we will come to this in a moment. But in general, the main function of gamma delta T cells is to eliminate infected and malignant cells in the human body. And they do so by recognizing different targets, recognizing the target cells in an MHC-independent manner. So the gamma delta TCRs, they recognize typically stress-induced ligands in target cells. And besides this recognition that does not require any antigen presentation, they also express several innate receptors, such as NKG2D, that enhance their functionality. So if we look closely at the two major gamma delta T cell subsets, starting from the B delta 2, these cells are preferentially located in peripheral blood. They express different cytotoxic functions, and they are able to modulate immune responses as well as present antigens. And these functions are triggered when they recognize in target cells different sets of stress-induced ligands, such as phosphoantigens. On the other hand, the non-B delta 2 gamma delta T cells, where we have the B delta 1 and delta 3, they are preferentially found in tissues such as epithelia. Similarly to the delta 2s, they express different cytotoxicity functions, and they can also modulate immune responses. And on top of that, they have very high tumor infiltration capacities. They are able to recognize different stress-induced ligands in target cells, but these sets of ligands are completely different from the ones recognized by the delta 2s. For example, some of these ligands can be expressed in MHC-like molecules. So considering all these biological features of gamma delta T cells, we can somehow understand why these cells can be relevant for immunotherapy. So as we have just seen, first of all, they can display multiple cytotoxicity mechanisms from their innate properties, but also by implementation of certain genetic modifications, such as the implementation of chimeric antigen receptors. So far, gamma delta T cells, they have shown a safe profile in clinical studies with low risk of cytokine release syndromes and neurotoxicities. And this safe profile is extensive to the use of the cells in allogenic setting, as they offer low risk of graft versus host disease and can be also produced from multiple cell sources. So ultimately, these cells offer an off-the-shelf potential for the establishment of an immunotherapy, as they offer the potential to treat multiple patients in a more cost-effective manner. Looking into the clinical landscape, it is of note the increase in number of clinical trials involving gamma delta T cells in the last years. Particularly last year, it was highly remarkable, this increase in clinical trials. The target indications for the use of gamma delta T cells are particularly solid tumors, but it's also of high interest to use these cells for the treatment of hematological malignancies, and it is also increasing the interest in the use of gamma deltas to treat autoimmune as well as infectious diseases. If we consider the adoptive transfer of the cells for a therapeutic approach, the main cell source used so far is allogeneic. And also, if we look at the trends from the preclinical work that is being done, there is a high interest in engineering the cells to confer them even higher functionalities and more targeted functions. So the development of a gamma delta T cell therapy, as now any other cell therapy, is regulated as a drug product. And as you probably know, the journey from the target discovery up to commercialization of cell and gene therapy can be long and involve different phases. And during these phases, the manufacturing as well as the analytical processes, they are also being shaped and they go through all the different stages. So this is just a simplified version of the complexity of the journey. And the point here is that the more the cell therapy advances through these phases, the less flexibility and room for change there is in the manufacturing and analytical processes. Therefore, it is very important to think early on in your development process, think translational on how you want to establish your manufacturing and analytical processes, particularly within the preclinical phase, to avoid having to perform requalifications and revalidations of your processes. From Miltenyi Biotec's side, we are committed to enable a smooth translation from the preclinical phase into the clinical one. Here we see several steps that can be applied in the manufacturing of a gamma delta T cell therapy. And here, this is an overview of the translational products that we have at the moment for the preclinical research, as well as the ones that can be used in the clinical manufacturing, including also different analytical tools, mostly based on flow cytometry, to complement for the analytical assays that have to be performed. So this is just the example of the current products that are translational. We have more products that can be used for the preclinical research, and we are working on increasing the portfolio to enable even more solutions also for GMP grade counterparts, particularly for cell separation. So now I would like to redirect the focus into some challenges for gamma delta T cell manufacturing that also lead us to the key considerations. So until now, the field has experienced two main challenges to establish the manufacturing of gamma delta T cells. First of all is the fact that since the cells are a rare population in general, expansion it is required to achieve sufficient numbers for clinical applications. However, there has been a lack of reproducible and scalable expansion methods. And on the other hand, gamma delta T cells have been known up to now to be difficult to genetically modify, So this is a challenge that remains, but the trend from the field is to implement genetic modifications. So this needs to be solved as well. Now, if we focus on the establishment of a process to manufacture gamma delta T cells, before we start, there are two key considerations or two main factors that we need to keep in mind when establishing our manufacturing process. And these are the cell source that we want to use and the gamma delta T cell subset that we want to focus on. If we look at the cell source, we can use either an autologous cell source, particularly peripheral blood or tissues, mainly tumors. Or also, we could use allogenic cell sources. Here, the range is wider. We could use some peripheral blood, cord blood, or even pluripotent stem cells. And the point here is that the cell source that we are going to use is going to determine the sample preparation and the cell separation strategies in our manufacturing workflow. And if we are working with an allogenic cellular starting material, this may also require the establishment of a differentiation protocol, like in the case of working with pluripotent stem cells. And in general, allogenic starting material would require the removal of alpha beta T cells and B cells to reduce the risk of graft versus host disease and B cell related disorders. Next, regarding the gamma delta T cell subset, this is also very important to keep in mind because the two main subsets that are being currently explored for therapeutic applications, which are the B delta 2 gamma delta T cells and the B delta 1s, they have different activation stimuli. So on one hand, the gamma delta, the B delta 2 gamma delta T cells, they become stimulated by the use of phosphoantigens, either natural or synthetic. They can be also stimulated using bisphosphonate drugs, such as zoledronic acid or pamidronic acid, together with accessory cells. And of course, feeder cells could lead to the activation of B delta 2. And then for B delta 1s, they can be stimulated using anti-B delta 1 agonistic antibodies, as well as feeder cells. And of course, because the cells express the CD3 molecule, we could use more generic stimulation via anti-CD3, stimulation with or without anti-CD28 co-stimulation. However, about this approach, I would like to remark that when using, so this approach also activates alpha beta T cells. So when using an allogenic starting material, this could be of risk. And besides the stimulation via TCR, of course, the cells also require specific cytokine support, like the cytokines you see here. Therefore, each gamma delta T cell subset has different requirements for activation, and this is then influencing, determining the expansion strategy that we need to establish. Therefore, the manufacturing workflow needs to be tailored according to the cell source and the specific subset of interest. So based on these key considerations, we have established a first manufacturing protocol focusing on the B delta 2 T cell subset. And once we have defined our subset, and we also decided to proceed with the use of allogenic starting material, in our case, peripheral blood, we aim to tackle the first main challenge in terms of manufacturing, which is the lack of reproducible, scalable expansion methods, by implementing an automated and scalable manufacturing workflow. And we did so by using the CliniMACS Prodigy platform. The CliniMACS Prodigy platform is an integrated cell processing platform that reduces complexity and footprint in the manufacturing procedure. It is an automated platform which helps with the process standardization. The platform and the manufacturing is implemented in a closed system to help increase the safety and robustness of the process. The platform is also versatile and flexible. So today we are going to focus on discussing the manufacturing of gamma delta T cells, but the platform enables multiple applications for all the cell types, and also enables the adaptation of different parameters during manufacturing protocol. And finally, the use of this platform is scalable and helps to simplify the technology transfer. So with the use of CliniMACS Prodigy, we have been able to transfer all the steps for the manufacturing of gamma delta T cells in this one closed system. And basically, the workflow that we have established in CliniMACS Prodigy enables to speed up process validation, as well as to achieve clinically relevant doses that ultimately help to accelerate the access of the therapy to patients. And this is possible because the workflow is scalable and automated. It enables high expansion rates of the B delta 2 T cell subset with efficient transduction. And the workflow also comes together with different certified trainings for any user to master the handling of the complete workflow. And also we provide technical and regulatory support. So now we can have a look at the different steps that we have established in this manufacturing workflow. As I mentioned earlier, we use allogeneic leukapheresis as starting material, non-mobilized and fresh. And after the sample wash, we implement a step to remove alpha beta T cells as well as B cells. This step, we have implemented it to ensure the safety of the product, since we are using allogeneic starting material. Once we have depleted alpha beta T cells and B cells, we start activating the gamma delta T cells in the remaining cell fraction with the use of zoledronic acid and different cytokine support that induces the preferential expansion of the B delta 2 T cell clones. And with the use of an optimized activity matrix in the Prodigy software, this is leading to the high expansion potential of the cells, and also includes several parameters that can be adapted by the user if needed. Transduction is an optional step, but we have also defined a specific activity matrix to conduct an efficient transduction with a particular lentiviral vector that I will explain in the next part. And finally, we can harvest our cell product consisting of highly pure expanded B delta 2 T cells. In this slide now, we can have a look in detail at all the material that is needed for this manufacturing workflow, as well as material needed for the in-process control and release testing, or shortly IPC-RT. As I mentioned, we start the process performing depletion of alpha beta and C19 positive cells. We are using a particular process, and some of you might be familiar with this process in general because it was established long ago, particularly to process hematopoietic stem cell transplant to increase the safety and the anti-tumor efficacy of the stem cell transplant by depleting alpha beta T cells and C19 T cells. But now we have a new process to conduct this step. We have a new application, which is the one we are using in our gamma delta T cell manufacturing protocol. And once we are depleting the cells using this application, their remaining cells are transferred into a different tubing set with the use of a different application, shortly called TCT-LS, with the use of a specific activity matrix, with or without the transduction activity, depending on whether this is needed or not. So once our leukapheresis product is depleted from alpha, beta, and C19 positive cells, we proceed to the activation of gamma delta T cells with the use of zoledronic acid in combination with IL-2 and IL-15 cytokine support in our TexMAX GMP medium. Now in this slide, we can have a look at some exemplary data that we obtained during our verification runs. Here you see the efficiency of our depletion step. So after alpha, beta T cell and B cell depletion, the majority of the cells remaining are actually monocytes and NK cells, which we actually need for our expansion protocol using zoledronic acid. And of course, we have a slightly increased enriched fraction of gamma delta T cells. This depletion process overall is really efficient. As we can see here, we obtain a logarithmic depletion value higher than 4 for alpha beta T cells and higher than 3 for B cells, which is very significant. It's a very stringent depletion step. However, even though the depletion step is very stringent, we are able to recover to obtain a very high recovery of gamma delta T cells of approximately 90%. And within the gamma delta T cells that we recover, approximately 72% are B delta 2, the B delta 2 subset. And they possess high viability after this step. So once we have separated the cells and we trigger the expansion, we keep them for 14 days in culture. And after these 14 days, we recover a highly enriched gamma delta T cell final cell product. So there is a remarkable expansion of gamma delta T cells, as we see in the graph on the left. Among those gamma delta T cells, 98% correspond to the B delta 2 subset, and they are highly valuable. And besides that, they also express a favorable phenotype of effector memory by day 14. Here, I just wanted to give you some numbers as a reference. We start the cultivation of the cells using a total of 1 billion total white blood cells. And by day 14, when we harvest the cells, we obtain on average approximately 7 billion total gamma delta T cells, which means we are, with this manufacturing protocol, we are able to achieve a fold expansion higher than 200. So this is really high numbers, but what would they mean in a clinical setting? So if we look at some examples of doses that have been applied in recent clinical studies, if we take as an example, particularly the dose that is mentioned here and this particular patient weight, with our manufacturing protocol, we are able to generate approximately 20 doses from one single non-mobilized leukapheresis, which is really remarkable. However, we are able that in certain instances, even higher cell numbers of gamma delta T cells may be needed. So here we have thought of different upscaling options. On one hand, it is possible to connect the CliniMACS Prodigy platform to other external culture systems to enhance or to upscale the expansion of the cells and then bring the cells back into Prodigy for the final formulation. This is already possible via a customized application program. And on the other hand, we can also think of using the CliniMACS Prodigy to generate a small batch and automate this step, and later on thaw aliquots and perform the larger scale expansion of the cells. So ultimately, the point with these upscaling options is that the Prodigy platform can be the key and the centerpiece of these upscaling processes, since it enables the automation of the critical steps in the process, such as are the separation or the depletion of alpha beta T cells and B cells, as well as the activation, the triggering of the expansion and engineering. But we are also aware that in certain instances, some specific needs need to be implemented in a manufacturing process. And for that, we are also able to allocate or to implement customization at any stage or step of the manufacturing process, as you see here, for the preparation of the sample, the separation of cells, transduction, cell culture, and the final harvest. So if this is of interest, we can provide a service to customize the process according to specific needs. And now I would like to move on to the next part of the session to discuss how we can implement efficiently genetic modifications into gamma delta T cells. So we saw at the beginning, this is a challenge that remains. There are different tools to implement genetic modification of the cells, and lentiviral vectors is one of the most common strategy to include genetic modifications into immune cells. However, so far, not many lentiviral vectors have worked very well to modify gamma delta T cells. But we were able to identify one particular pseudotype, the baboon pseudotype lentiviral vector, that can work really efficiently transducing the cells to implement genetic modifications. And we looked into this when we first started to test and assess how we could modify our gamma delta T cells. And of course, we started using the VSVG lentiviral vector, which is the most common one to be used to implement genetic modifications. And we compared it with the baboon pseudotype 1 with the use of different CAR constructs, as you see in these images. So with VSVG, the transduction efficiencies were really low. There was really low expression of the CARs on the gamma delta T cells. However, when we were using baboon pseudotype lentiviral vector, the transduction efficiency increased significantly, and we were able to observe this with the use of different constructs, like the ones mentioned here, V7H3, C19, and C33. Moreover, we also analyzed the killing efficiency of target cells using the modified transduced cells, either with VSVG lentiviral vector or baboon lentiviral vector. And as you see here, only when we use a baboon lentiviral vector, we were able to see a significant target cell killing at different effector to target ratios. So this work was done by our R&D team and has been recently published. So if you are interested in learning more details, I invite you to look at this publication. So this protocol was established for a small scale manual process, and now we have transferred this protocol into our CliniMACS Prodigy platform. And this is how it goes for to transduce gamma delta T cells. So again, we deplete alpha betas and CD19 positive cells from our leukapheresis sample. We activate the cells for 3 days, and on day 3 we include our vector together vector fusing 1, which increments the transduction efficiency in the process. And we keep the vector together with vector fusing for 24 hours, and on the next day, on day 4, we wash them out, and then we continue with the expansion phase up to day 14. So using this protocol, this is some exemplary data that we have obtained. As you see here, on average, we obtain around 60% of CAR gamma delta T cells by the end of the expansion protocol. And because we transduce the cells on day 3, and there are still also quite some NK cells left in the sample, this also kept nicely transduced. But this is totally fine because the NK cells, they also, similarly to gamma deltas, they recognize target cells independently of MHC molecules, so they don't represent a risk for graft versus host disease. And if so, they can actually enhance the functionality of the final cell product. And more importantly, we not only get a very high transduction of the gamma delta T cells, but we also have a very safe profile. When we look at the vector copy number, this remains below 5 copies, which is within the requirements, the regulatory requirements. So again, also for this transduction protocol, we start from 1 billion of total white blood cells. And we obtain approximately 60% transduced gamma delta T cells with a very low MOI. And so far up to now, we were able to harvest approximately, on average, 1.5 billion total gamma delta T cells. So at this point, you might be asking yourself how you can access this baboon pseudotype lentiviral vector. And I would like to explain that there are different pathways that you can follow to access this vector. On one hand, if you have a particular construct of interest that you have designed, we can integrate this construct in the baboon envelope lentiviral vector backbone and produce this vector for you, either in PD grade or GMP grade. And besides that, we also offer a GFP expressing baboon envelope pseudotype lentiviral vector that can be used to perform proof of concept studies. So both options are a service provided by Miltenyi Bioindustry as of now. And on top of that, we are working on enabling very soon the access to an off-the-shelf vector, which is the C19 baboon pseudotype lentiviral vector, also both in PD and GMP grade, to complete our lentiviral vector offering. So if you are interested in learning how to access this, about the different possibilities, please contact your local Miltenyi representative for more information. And last but not least, I would like to go over some flow cytometry tools for the analytical testing of the manufactured gamma delta T cells and their phenotyping. So we have just learned how we can optimally manufacture gamma delta T cells, particularly the V delta 2 subset, using CliniMACS Prodigy platform, and different steps, how we perform them in this workflow. But of course, the manufacturing process goes hand in hand with the analytical procedure in order to finally qualify the final cell product. So for these, different assays have to be performed for in-process control and the release testing of the product. Here you see some examples of certain assays that can be performed, such as the analysis of the cellular composition at different stages of the process, or the evaluation of the efficiency of the genetic engineering, such as transduction efficiency. And many of these assays, they can be performed by applying flow cytometry tools. And here I would like to introduce you our MaxQuant analyzers that can be a great fit into this analytical process. So MaxQuant analyzers, these flow cytometers, they can truly help to standardize flow cytometry in cell manufacturing. And they do so in different ways. On one hand, They can implement the express modes. Express modes, they are a software tool that enables the automated sample acquisition, data analysis, and reporting. At the same time, mass quant analyzers can implement or have implemented the smart gain technology that enables a consistent and reliable assay transfer to different instruments, which makes it easier to scale up and to transfer a method across different sites. The software to manipulate the mass quant analyzer is 21 CFR part compliant, which helps with audit trail and also in a control user management system, as it enables the generation of analysis reports with e-signatures. And of course, these flow cytometers, they are not alone. They go hand in hand with different standardized reagents, such as the reaffinity recombinant antibodies, to enable reproducibility and the standardization of the flow cytometry assays, and also our cut detection reagents and MSC maximers that were recently released. And we make use of all these different reagents to create some predefined antibody panels that we verified and we can provide as well to facilitate the in-process control and final release testing. So about that, we have developed such panels for the manufacturing of gamma delta T cells. And as you can see here, these panels enable, on one hand, the determination of the cell count and the viability of the cells, but also the analysis of the cell composition at the different stages of the manufacturing process, particularly to determine the depletion efficiency of the alpha, beta, and B cell depletion step. And of course, in case transduction is implemented in the process, we also have a specific panel to conduct the evaluation of the transduction efficiency within the process. And furthermore, because gamma delta T cell phenotyping can be also of interest during manufacturing or during the preclinical phase research, we also offer several tools for the analysis of the main subsets. So we have different antibodies to identify the B delta 1, B delta 2, and B delta 3 chains in the gamma delta TCRs. And of course, besides these TCRs, we offer many other antibody conjugates to identify and determine other markers of interest for phenotyping. And with this, I'm coming to the end of the presentation, and I would like to finalize with a few take-home messages. First of all, I would like to remark again that the cell source and the gamma delta T cell subset of interest determine the manufacturing protocol and really need to be considering when setting this up. The manufacturing workflow that we have established for B delta 2 T cells using CliniMACS Prodigy already enables the generation of sufficient numbers for clinical applications. And the use of mass quant analyzer and the ready-to-use panels can facilitate the flow cytometry analytical testing of gamma delta T cells. So overall, the combination of CliniMACS Prodigy and mass quant analyzer is the perfect match that enables to speed up the process validation of your gamma delta T cell manufacturing protocol and ultimately accelerate the access of your gamma delta T cell therapy to patients. So with this, we came to an end, and I would like to thank you for your attention. Thank you so much.